Hello, my cutest little champions. I hope you're doing really great. And I welcome you all to Masterstroke series, where today we are going to deal with the chapter Biotechnology and its applications. So what do I need from you? One NCERT and one pen. Whenever I say, hey, this is something that is not usually asked, or basically this you might not predict coming in the exam, but it will come. So all these things, when I say no, you can just write it there. Okay, this type of questions can be coming around. So we should be really careful. We should be studying this particular thing. You get it? So let's go ahead with the first thing. Okay, so your chapter starts with the first thing, that is about different type of engineering, genetic engineering, things like that. Then they land on to tissue culture. Okay, out of nowhere. So tissue culture, my dear children, and micropropagation is basically done in the case of plants, where what you're doing is that in tissue culture, you take a part of the plant, okay, certain cells, meristematic cells, which has the ability to divide, and then you're going to put this particular plant into some medium that has enough growth hormones and stuff like that, so that basically they can divide, and then they can give rise to root and shoot and everything, and then can give rise to the whole plant. Now, growing a whole plant from an explant means any plant part in a sterile condition is known as tissue culture. Now, explants are placed in special nutrient medium for what? Growth. Now, one very important property about the cells that are there in the explant, that is meristematic cells, they are totipotent. What do you mean by that? That means from a single cell, they have the ability to regenerate a whole plant. That is, it can give rise to leaves, roots, whatever they want. So, it is known as totipotency, when you have the ability to give rise to whatsoever you want. Okay, and a single cell has that ability, it is known as totipotency. So, this particular ability is being used in tissue culture to generate a whole new plant. Now, micropropagation is basically the same thing, okay, a part of tissue culture, but producing thousands of genetically identical plants, and it is known as soma clones. All of them are clones, my dear children, and it is used for commercial mass production of crops like tomato, banana, apple, and things like that. Now, ma'am, why are we talking about tissue culture over here? Is there any need of it? Well, to answer that, before answering that, I have one more question. Okay, that is, can we generate virus-free plants? If we are generating like this, can we actually generate virus-free plants? Yes, we can generate. How? By taking the apical and axillary meristem. So, apical meristem, you know, where do they find? At the tip of the plant, my dear children, you have apical meristematic cells. Okay, now axillary buds, as in the buds that will be giving rise to what the stem, the branches and all, they are known as axillary buds. So, over here, the axillary meristem and the apical meristem are free from viruses. Even if the plant is affected by a virus, these meristematic cells would be free from viruses. So, I can take those cells to give rise to what? A healthy plant. Okay, and this has been done in plants like banana, sugarcane, and potato as well. So, this is a question, for getting virus-free plants, where should we get the meristematic cells from? From the apical or axillary meristem, first thing. Okay, now, so we were talking about tissue culture and things like that, and one more thing, let me explain that also. Okay, so all this method is basically giving rise to plant, giving rise to multiple copies of plants, or making clones of plants, my dear children. Okay, in tissue culture, you are taking explant and making different plants, and or basically from an explant you are growing a new plant. In micropropagation, you are making multiple plants. Now, somatic hybridization, where basically what you are doing is that you are removing the cell wall. You know, plant cells, they have a cell wall as well. So, you remove the cell wall. When you remove the cell wall, what are you going to get? You're just going to have what? The cell membrane and the content which is there inside. So, cell walls of the plants are digested to obtain naked protoplast. So, whatever is left behind when you remove the cell wall is now known as protoplast. My dear children. Now, what do? Protoplasts from different plant varieties with desirable traits are fused. They are made to fuse. Now, what will it give? It will give hybrid. So, fused protoplasts develop into new hybrid plant. Somatic hybrids are the names. Okay, the resulting plants are known as somatic hybrids, because we are taking somatic cells and we're just fusing them. Here, there is no gamete formation and things like that. Pomato is an example where there is a fusion of potato and tomato and taking place over here. But it was not commercially successful. So, all the things that we saw until now, the tissue culture, somatic hybridization, everything, to an extent it has been a successful procedure. Tissue culture is still being used and things like that. But we needed something else. Something else which is less time consuming, something else that can for sure give us better products and things like that. And what was that something, my dear children? It is genetically modified crops. Is there any better mechanism for the production of better crops? Yes. So we have genetically modified organisms. Organisms as in it includes plants, bacteria, fungi, everything which is genetically modified. When we're talking about crops, we call them genetically modified crops. And how are they better than everything else? Because they would be tolerant to abiotic stresses. For example, resistance to cold, drought, salt, heat, everything. Okay, we are going to put certain genes. We are genetically going to modify them. We are going to put some genes. We are going to remove some genes in a way that they are able to survive in abiotic stress conditions as well. When there is too much high salinity, they will still be able to survive. When there is too less water, they will still be able to survive. Now, pest resistant varieties were also made. It reduces the need for chemical pesticides. You know, pesticides are being put, but we do not need pesticides now. In some plants, genetically modified plants, we do not need pesticides. Why? Because these organisms, these plants are going to release some toxins. So whenever a pest will come and eat it, the pest will be dead. It is not going to cause trouble in us, but to the pest. You get it? So pest resistant varieties of plants were being able to produce with the help of genetic modification. Now it also reduced post harvest losses. So after harvesting also there are losses. So it basically, genetically modified crops are the one that had improved storage and shell life. So it was able to survive even after harvest. It with less moisture, with so much moisture and things like that, it was still able to survive because of the changes that was being made. Okay, now efficient mineral usage. Genetically modified crops were good because they were using the minerals efficiently. So it's not like in normal plant, what will happen, it will take in all the nutrients, my dear children, really quick. So it will make the soil infertile. But in the case of genetically modified crops, they are going to take up the nutrients really slow in an adequate amount because they are able to survive with that amount of mineral nutrients which are there. Which is why there is efficient mineral usage, and in this way the soil will still be remaining fertile for other plants also to get generated. Now the most important point, enhanced nutrition. The nutritional value of the plant is going to get increased. Have you heard about golden rice? Well, golden rice is a vitamin A enriched rice. This was made in countries where the vitamin A deficiency was prolongedly seen, the eyesight problem and everything developed in Africa and all. So those countries know this genetically modified golden rice was being put up. Okay, why? Because the staple food is rice. If people are having rice and getting vitamin A, what else do people need? You get it? So it was vitamin A enriched rice. What is it, children? Golden rice, and this is a genetically modified crops example. Okay, cool. So this is something that I wanted to tell after so long of explaining tissue culture and somatic hybridizations. I hope you got what is tissue culture. Questions come on tissue culture also. I told you, right? How do we get virus-free plants with the help of tissue culture? So basically, take the cells from the apical or axillary meristem and then do the tissue culture method. You will get virus-free plants. Now, somatic hybridization, does it always work? What does it mean? So basically, it is when the cell wall is removed and the protoplasts are fused. That's how you get new hybrid types of plants, my dear children. Example is tomato, as I told you over here, but it was not commercially very much successful. Then came what? Genetically modified crops, which are over here, which are not over here, basically the advantages of which are over here. One very important example is also being put up over here, that is of golden rice. Cool. Now we will see other examples of biotechnology in agriculture. Okay, we were talking about plants, we'll continue talking about plants. So have you heard about BT crops? BT crops are really important. Why? In BT crops, what is happening, my dear children? First of all, what do you mean by BT? BT stands for Bacillus thuringiensis, which is a bacteria. Okay, BT stands for Bacillus thuringiensis, which is a bacteria. Now it is going to act like a biopesticide. What do you mean by biopesticide? Basically, you do not need pesticide to kill a pest. Instead, if Bacillus thuringiensis is there, it is going to kill all the pests around there. It is not going to cause a problem to the plants as well. How? Basically, Bacillus thuringiensis has a gene in them which is known as cry gene, my dear children. Has a gene in its body, that is in its genetic material, that is known as cry gene. And cry gene, it codes for a toxin. It codes for what? A toxin. Now this toxin is basically in an inactivated condition inside the bacteria. Inside the bacteria, it's an inactivated condition. Suppose I am a butterfly caterpillar. Okay, I came and ate the plant which was having the bacteria, that is Bacillus thuringiensis. What will happen? Now the bacteria is inside my body, inside my gut. In the alkaline medium, in the alkaline pH, my dear children, in my gut, if I am an insect, inside my gut there is an alkaline pH. In this alkaline pH, the inactive toxin which was there becomes active, and it is going to cause damage to the cellular layers, epithelial cellular layers of my midgut, and I am going to get killed. So what if what if we take the same gene? We do not obviously expect the bacteria to be always there. So what if we just take the gene and put it in the plant's body? The plant will also produce the same toxin. Now when an insect will come and eat the plant, what will happen? The insect will be dying. Now don't you think it's a good mechanism? Yes. So wherever this gene was put down, now you call them BT crops. We call them BT crops. For example, BT cotton, BT rice, BT tomato, BT brinjal, lot of things were being made with the help of this. Remember, Bacillus thuringiensis is a biopesticide or a biocontrol agent. Okay? Works. Now, BT cotton mechanism. So, BT produces insecticidal protein. It targets specific insect pests. Okay. Now, toxin exists as inactive protoxin. It exists in the bacterial body as inactive protoxin. It does not harm the bacteria, please remember that, because it is inactive. Now, insects ingest the protoxin. Insects which are there, they are going to ingest it. Now what will happen? It is activated in the alkaline gut pH. In the gut, it becomes activated. Where exactly? So activated binds to the mid-gut epithelial cells, creates spore leading to cell lysis and insect death. You get it? It is going to bind to the mid-gut. In the mid-gut region, this is very much important. They can ask you which portion of the gut, foregut, this gut, that gut. So basically it's mid-gut, and what are the cellular layers? It is epithelial cells where it is going to cause the trouble. It is going to make pores, and this will lead to cell lysis and the insect death as well. I hope this particular thing is clear to you. Now you have BT genes and the target pest. So genes, I told you it's cry gene. You have to know which cry gene. Cry gene obviously releases toxins, and now these toxins are also specific to certain insects. For example, Cry1AC and Cry2AB is for cotton bollworms. Is for what? Cotton bollworms. How do I remember it? Listen to me. Cry1AC. So the first letter starts with C, so cotton. Okay. Now Cry2AB. Second letter starts with B, so it is bollworm. So cotton bollworm, you have two things, that is Cry1AC and Cry2AB. Okay, Cry1AC and Cry2AB. Now you have Cry1AB. Now you might think cotton bollworm and corn borer both have CB, ma'am, then how? So here two things are involved. Cotton bollworm is also long word, so you can think like that. Now corn borer, my dear children, Cry1AB. So over here C for corn and B for borer, first and the last letter. Now there is only one word involved, that is, there is only one thing involved. So Cry2AB anyways is being utilized. Over here you just have one AB left, and that is for corn borers. So different cry genes, as I said, target different insect groups. For lepidopterans such as tobacco, budworm and army worm, there are different. Okay, for coleopterans like beetles, there are different. For dipterans like flies and mosquito, there will be different. So there are different types of organisms, groups where it is going to target: lepidopterans, coleopterans and dipterans. Cool. Please remember the cry gene, please remember the name of that guy, and also biopesticide or biocontrol agent. It is there in inactive protoxin state and gets active in the mid-gut and it damages the epithelial cells. And the name of the cry genes and what does it do? Cotton bollworms, corn borers, very famous question. So this is the picture. Cotton ball destroyed by bollworms, that is A, and fully mature cotton ball, which is there. Okay, this is the actual one, this is the destroyed one. Okay, going ahead. What do we have? We have RNA interference, that is the next thing that we have. So, biotechnology in agriculture, we have RNA interference. What do you mean by that? So, before getting ahead with anything, I want to explain you something, that is, you know, in our body we have DNA. This DNA is going to transcribe into RNA. In which form? Basically in mRNA form. mRNA. Now this mRNA is going to get sandwiched between the ribosomes, and that is how translation happens. That is how translation happens. But have you noticed one thing? The mRNA is single-stranded, my dear children. This is a single-stranded RNA. mRNA just has a single strand. Suppose what happens? Somehow it's made to be double-stranded. Will it undergo translation? It would not undergo translation. Hence, will it make the proteins required? No proteins are going to be made. And this exact mechanism is being used in RNA interference. This method is used in RNA interference. You are interfering between RNA translation, my dear children. What exactly happens is that, okay, there is a small little worm, cute little worm, not very cute, but yeah. Okay, that is Meloidigyne incognitia. Now, this particular worm which is there, it is going to affect a plant, that is tobacco plant. Okay, it is going to affect the plant, that is tobacco plant, mainly what? The roots of the tobacco plant. Fine. So, basically, it was producing certain proteins and everything which was damaging the plant and everything, okay. So we want it to stop the production of that particular protein. How we can do that? Basically, we can take the gene from here, we can take the gene from Malardina and Incognitia that is coding for that particular protein, and we are going to put it inside the bacteria. Inside the bacteria, that is Agrobacterium tumefaciens. Ma'am, why are we including a bacteria over here? I'll tell you. So this DNA, whatever is there, this gene has been inserted into the plasmid of Agrobacterium tumefaciens, which is known as TI plasmid, tumor inducing plasmid. If you remember, you remember. So Agrobacterium tumefaciens is the one that usually causes tumor in the case of plants. But if we remove the tDNA, it's known as tumor inducing plasmid, and it has a segment that is known as tDNA. If you remove the tDNA and if you put a gene of interest, my dear children, it's not going to harm us. Still it thinks that I have the tDNA, it will still go infect the plant, but what is it actually going to do is that it is just going to transfer the DNA which is there, but it does not know that it is not transferring tDNA but our gene of interest. So the gene which was here, the DNA which was here is now in the plant. It's now in the plant. Okay, now what has happened is that basically this particular plant is going to make double-stranded RNA out of it. How? What? Double-stranded RNA is being made out of it. Cool. Now what will happen? When it is forming double-stranded RNA, what is going to happen? This double-stranded RNA, my dear children, when this guy has or eats a part of the plant, what is happening is that the double-stranded RNA is going to get inside the body of this organism. Now, this organism has its own mRNA which is going to get translated into the protein. But since now it has double-stranded RNA, what will happen? This particular strand is going to form a bond with this mRNA and makes this mRNA as double-stranded RNA. Now will it undergo translation to form the protein? No, it is not going to do that. So aren't we interfering? We are interfering. Now we will study. So over here, nematode problem. Malardina incognitia infects the tobacco roots, reducing the yield. Now nematode, it's a nematode as in it's a Hask helminthes, okay. Now RNAi strategy. So silences specific mRNA to prevent nematode infestation. Now listen to me, RNA interference is a natural technique, okay. It is a very natural thing that happens in the eukaryotic body. It's a mode of cellular defense in the case of eukaryotes, not prokaryotes. This is again a very important question. Which among the statement is wrong? Which statement among this is wrong? Sorry. So basically RNA interference takes place in eukaryotes and prokaryotes. Wrong. Only in eukaryotes, and in eukaryote it acts as a cellular defense. So something comes in, we want to mute it, so we do RNA interference. Now mechanism, so double-stranded RNA formation. Complementary RNA binds to the target mRNA, blocking the translation. That is how it works. So agrobacterium vector used to introduce nematode-specific genes. The agrobacterium vector is made to introduce the nematode-specific genes into the host plant. Now the host plant produces sense and antisense RNA, forming double-stranded RNA. Listen, the sense, the complementary RNA is known as, one is known as sense and the other one will be known as antisense. Very important, again a very important question. Okay, that is how a double-stranded RNA is formed. So host plant would be producing what? Sense and an antisense RNA, that is a double-stranded RNA. Now this double-stranded RNA triggers RNA interference, silencing nematode mRNA. Nematode cannot survive, making the transplant what? Resistant. It will make the plant resistant. Since it is not able to produce the proteins, it won't be able to survive, my dear children. It won't be surviving, it won't be infecting, and it will just die off. Cool? Works? So this is how it works. Okay? Now, RNA interference, I hope it's clear. Please remember double-stranded RNA, also agrobacterium vector. Always for plants, if you want to transport something, if there is a vector, that is agrobacterium. Okay. Now, sense and antisense RNA, very, very important piece of information. Okay, going ahead. We have biotechnology in medicine. So, insulin production, you know what? Previously, we were taking insulin from the cattle and the pigs because we had some similarity with them. But the problem is that some people were getting allergic to this. So, obviously, can we continue doing so? We cannot continue doing so. So, what can we do? Basically, we can take our gene and make some other organism produce it, and then we can use the same product, that is what we are going to get from them. That is what something that we can do. So insulin was extracted from cattle and pig pancreas initially, but animal insulin causes allergies and immune reactions. Now what? Human insulin structure. Please remember. So if you see human insulin, my dear children, you have A polypeptide, you have B polypeptide, and this thing that you see, this is known as C polypeptide. Now this is pro-insulin. If you can see, this is what? Pro-insulin. This is not the actual hormone, this is inactive hormone. To make it active, you have to remove the C polypeptide from here. You have to remove the C polypeptide from here. That's when A and B will come together, form disulfide bonds and become what? Active insulin. So pro-hormone, human insulin structure, it consists of two polypeptide chains A and B linked by disulfide bonds, as you can see over here. Okay, now pro-hormone form initially produced with an extra C peptide which is removed during what? Maturation. So in our body, this C peptide is being removed. Okay, but if I am putting the gene for human insulin into E. coli and expect it to cut the C polypeptide as well, is it possible? No, because it does not have the same machinery as us. So I cannot do that. Then how can I make my production of insulin over there? So when people were confused regarding this, there came Eli Lilly company in the year 1983. In the year 1983, who came? Eli Lilly company, who said that. It's not like they were not there before, they were there. So they said that we have a solution for it. What was the solution? Basically, why don't you take the gene separately? The genes coding for A polypeptide, take it separately. The gene coding for B polypeptide, take it separately. And then put both the genes into plasmid and then put it into the E. coli. E. coli will be producing A polypeptide separately, B polypeptide separately. What we can do is we can extract it from them and we can join them under laboratory condition. The problem is sorted, right? So they created two sequences, A and B genes for human insulin, introduced them into E. coli plasmids for separate production, not same production, separate. They were extracted and combined A and B using the disulfide formation. Okay, advantages was large scale production of human insulin using bacteria was done, and this was known as humulin, my dear children. What is the name for this? This was known as humulin. Is that clear? I hope it's clear. Okay, so C polypeptide is just there in the pro-hormone, not in the actual version of the hormone. So this is biotechnology in medicine. Now there is one more scenario of biotechnology in medicine, and that is gene therapy, my dear children. Just give me a second, I'll just reduce the temperature, it's too hot up here. Okay, in gene therapy, so purpose is correct gene genetic defects by inserting normal genes into patient cells. So first thing, my dear children, when did it happen? So in 1990, there was a 4-year-old girl with ADA deficiency. ADA stands for adenosine deaminase. Okay, it's an enzyme and this enzyme plays a major role in our immune system. Fine. So it is responsible for our immunity and things like that. But the child was not having this particular gene. There was a deletion of the gene that was coding for what? Adenosine deaminase. Deletion is also very much important over here. Fine. So what could be done in that case? So you have two treatment. One is bone marrow transplantation and enzyme replacement therapy. Bone marrow transplantation, you transplant the bone marrow, and bone marrow has the cells, right? You have the cells and basically you get it from someone else and these cells will divide, but it is not always curative. We cannot rely on that. Okay, and a kind of risk as well. So bone marrow has the cells, hematopoietic stem cells that will be dividing, giving rise to different type of cells. But again, that's not a surety how it will be happening in our body if you're taking it from someone else and putting it over here. Now there was enzyme replacement therapy where basically the enzyme was given. But again, you need repeated injections for that. In enzyme replacement therapy, you need repeated injections for that. So all this is also is not very much I say it's curative, but it's not completely curative because you need repeated injections. Then came gene therapy approach. Where was what done is that if I am a human being with adenosine deaminase deficiency, my lymphocytes were taken. Lymphocytes are a type of WBCs, okay? It's a type of WBC, white blood cells. So my lymphocytes are taken, and then what is done is that into my lymphocytes, a functional cDNA, complementary DNA, coding for adenosine deaminase was being put over here. Is it directly? No. Whenever you are infecting an animal cell, whenever you want to put something in the animal cell, you need a vector, and here the vector is retrovirus. In animal cell, it is always retrovirus. In plant cell, it is agrobacterium tumefaciens. Okay. So with the vector, you introduce complementary DNA over here that codes for adenosine deaminase. Now what will happen? This particular cell will have the efficiency to produce adenosine deaminase. Now then these cells are taken back and put into my body. But again, is it a complete cure? No, it does not give me complete cure. Why? Because I need to do the repeated infusions again and again and again and again. Because lymphocytes which are there in my body, they are not immortal. They will be dying. They will be dying. Other lymphocytes will be forming. They would not be having it. So that's a big problem, right? So that is also not permanent cure. What is the permanent cure for this? The cure is introducing ADA gene into the embryonic cells. If I have from the bone marrow, I have the adenosine deaminase gene, my dear children. From that cells I take it and I put it into the embryonic cells. In the embryonic stage itself, that's when I can completely cure it permanently. But then I have to know that this person has a deficiency for ADA. Got it? Then only I can cure it. So this is about gene therapy and everything. So gene therapy, as I said, it is not a curative approach. Over here if I do it, okay, over here if I put it in the embryonic stage, then it is fine. But apart from that, it's not. So there are 3 things given in NCERT that is not completely curative, but just there. But over here, one thing that is permanent cure, that is putting in the embryonic cell. Please remember, it's ADA deficiency. It is affecting your immune system. The lymphocytes are taken, put it back. Using the retroviruses we are going to insert the cDNA, complementary DNA, cDNA. Okay, that is going to code for adenosine deaminase and things like that. Done? Cool. So that is about biotechnology in medicine. Now there is early disease diagnosis as well. Again, use of biotechnology, let's see. So limitations of conventional methods. So serum and urine analysis cannot detect what? Diseases at an early stage. Obviously, we cannot detect by just a serum or a urine analysis. You need advanced techniques like what? Recombinant DNA technology, PCR and ELISA enable what? Early diagnosis. What is a very good thing about polymerase chain reaction or PCR? It amplifies even small amount of pathogen DNA for detection. So even if it's small, if we keep it for amplification, it will increase the number and we can easily detect it. That is the use of polymerase chain reaction, and this is a question that usually comes. Okay, which technique is used to basically detect even traces of pathogen DNA? So it is polymerase chain reaction because it amplifies it as well. Now used for HIV detection, cancer gene mutation identification and genetic disorder diagnosis as well. So people usually think HIV is just going for ELISA. ELISA is the only test. But here also, if there is even little thing up there of HIV virus, we can still detect it with the help of polymerase chain reaction, my dear children. Please remember that. Okay. Now we have DNA, RNA probes and autoradiography. What do you mean by probes? So these probes are nothing but the DNA or basically RNA, which might be associated with some radioactive thing. So it will be under UV light, it will be showing up its color and things like that. So what is the use of it? Suppose I know that this particular content DNA which is there, it will only go and bind to the complementary DNA which is there. Suppose this is only found in a person who has having one particular disease. If it is going and binding and making a complementary pair over there, that means that person is also having the disease. So radioactive probes hybridize with the complementary DNA. Mutated genes do not hybridize, allowing detection via autoradiography. So if or else, for example, I think that this is a normal gene, normal genetic code which is there. So if it does not go and attach, that also means that there is some mutation up there, which is why it is not coming and attaching. So that also shows up, and this can be viewed under autoradiography machine. Now there is something that is known as enzyme linked immunosorbent assay, that is ELISA. How does it work? It works on the principle of antigen antibody binding. I already taught you in human health and diseases chapter. Okay, how does it work? The basic principle is antigen antibody binding, my dear children. For example, suppose I know that in a HIV patient's body, these particular antibodies are being made in response to HIV. Okay, it's already there. It's already there. I'm not taking it from them. I already know it, so I have prepared over here. Now I am taking their blood sample. The antigens, if they come and bind over here, that means what? This particular person basically has HIV, which is why antigen is coming and directly binding to the antibodies which are only produced in response to HIV. But I cannot still say it. What do? I have another antibody which will come. Again, ready-made antibodies that I have will come and attach over here. But these antibodies are attached to certain enzymes, my dear children. So enzyme linked. Okay, it is linked to some enzyme, and to view it, what do? I put some substance over here. It will react and show up a color. Depending on the color intensity and things like that, I understand, okay, if the color is there, that means yes, the person is having what? This particular disease. So it's based on antigen antibody interaction. There are different types of ELISA, sandwich ELISA, this ELISA, that ELISA. Okay, so I'm not going in detail, but what is in the NCERT, that is being taught. So it detects infection by identifying antigens or antibodies in the patient's body, my dear children, either thing. Okay, so polymerase chain reaction, even for small amounts of pathogens, used for HIV detection, cancer gene mutation identification, genetic disorder, everything. DNA RNA probe method is also there. You can view it under autoradiography machines. And then you have enzyme linked immunosorbent assay, which works on the principle of antigen antibody binding. Done. Now, which of the following diagnostic tool allow for detection of very small concentration of bacterium or viruses by amplifying the nucleic acid? I told you. What is it? It is ELISA. Sorry, it is what? PCR. Small traces, it is PCR. Polymerase chain reaction, it amplifies it. 2021 question. Oh, children. So basically, have you already registered for KCET? If you have not, then you should be registering for not KCET. I was talking about Abhyas KCET test series. Yes. So if you have not registered, then it's high time to register, because the competition is really high up. But that does not mean you're not gonna win this game. You are definitely in the winning team itself. So do not be worried about anything. But as you know, we need much more amount of practice. Just like how much effort you're putting for understanding a chapter, that much effort is also needed for solving the questions. So we have 35 full syllabus and chapter wise tests, my dear children. Please make use of it. There is video solutions for all the questions. There is chapter wise one-shot videos. There is chapter wise insight for last 5-year KCET papers, and that is not a small thing, right? And for Lakshya KCET crash course also, you have daily practice tests, one-on-one doubt session, guided study hours. Now this is a crash course. This is totally different from KCET test series. Okay, this is online. This is totally online, and Lakshya KCET crash course is an offline thing. For 21 days, you have to be at our campuses. It's a residential course, so if you want to stay, we will provide you hostel facility. If you do not want to stay, come from home every day. If you're there in Bangalore, no problem, you can do that as well. Okay, but this is a 21-day course, and the teachers will be there, totally there to guide you out. So if you want any information regarding Lakshya, you can just call up this number, or you can click on the link in the description box. Apart from that, if you're talking about Abhyas, my dear children, again the link is given in the description box, and this is the detail. You can go ahead with this. Yes, please do not delay, because you know the time is flying, your exams are quite near. Yes. And now, transgenic animals, the second last topic of this particular chapter. So when animals are basically modified, you call them transgenic animals. Okay, so transgenic mice is being used mostly. It is 95% of the case, it is mice only. Please remember, this is also very important question that is put up over here. Rats, rabbits, pigs, sheep, cows, so many things are there. They are also transgenic animals. Transgenic as in, I am putting my gene into them and making them work like that. This is known as transgenic animals. Okay, whenever I am putting my genes into an organism to basically see its function or to produce something in them, okay, from my gene, that is also transgenic animals. Whenever I am doing an alteration in them, okay, in their genetic makeup, this is known as transgenic animals, my dear children. Now, application to study normal physiology and development. For example, it helps in understanding gene regulation and effects on growth, development and body function. Example, insulin-like growth factor research. For example, I put the insulin-like growth factor in their body and see what is the actual effect that it is being shown up there. So am I not able to understand the function of that particular gene? Yes. So am I not able to understand what this particular gene have to do with the growth, with the development and the body functions as well? Yes. So this gives me a total idea, and I can work around it. So for studying the normal physiology and development, I can do this. Okay, second thing, for disease study and human models. So obviously we and mice have so much similarity, almost like about 90% of our genetic material is being matched up, right? Do you know that? Well, it is yes. So in them and us, my dear children, obviously if you put our genes and see what all effects are being there, we can study different diseases as well. Okay, so if we mutate certain genes and then we provide treatment and see whether this treatment is working in them or not, is it curing the disease or not, we would be able to find out what cure for a lot of diseases. So it is created as models for human diseases like cancer, cystic fibrosis, arthritis and Alzheimer's. Please remember the names of these diseases: cancer, cystic fibrosis, arthritis and Alzheimer's. Okay, so all these things were being researched with the help of what? Transgenic animals, which are being put up over here. Okay, now biological products. For obtaining biological products also we can take the use of transgenic animals. So in this particular paragraph, this is the most important point. So you know what, in our body there is alpha-1 antitrypsin being produced. Alpha-1 antitrypsin being produced. This is a protein, and this protein is required for the treatment of emphysema, which is a respiratory disorder. Okay, so we want this alpha-1 antitrypsin to be produced in more amounts. So what do? We put this gene, we take the gene and put it in the organism, transgenic organism, and make them produce it. So it is used for human protein production, example alpha-1 antitrypsin, for emphysema treatment. Apart from that, if you have heard about transgenic cow, that is Rosie, in the year 1997, okay, this cow was made to basically produce the milk which is rich in the human protein, okay, human protein, that is alpha-lactalbumin. Alpha-lactalbumin. So there were certain changes being made such that the milk that it was producing had enough quantity of the human milk protein, that is what? Alpha-lactalbumin. So alpha-1 antitrypsin for emphysema and alpha-lactalbumin, which is a milk protein in humans, my dear children, was being produced by Rosie. So for biological products also you can take the use of transgenic animals. Now for vaccine safety testing, obviously on humans people would not be doing the safety testing. On animals which are kind of which have more genetic resemblance to humans, on them the vaccine safety testings were being done. Even though it's a really bad thing, I do not follow this, but how do we find the cure? That is the question. So no one has a solution for that, so that's the thing. So transgenic mice used for testing vaccine safety, example polio vaccine, before the human trials. Before putting on to humans, it was put on mice to see the vaccine safety. Similarly, chemical and toxicity testing. So for example, one particular organism is made to be sensitive knowingly, and then we put certain amount of chemical in them and see what is the reaction. If even after being sensitive the reaction is not too very much, that means it is safe for human use. If it is very much reactive, then obviously we cannot use it. You get it? So chemical and toxicity testing is also one such thing. So genetically modified animals used for quick and accurate toxicity test for drugs and chemicals, that is also one point that you should not miss. So there are 5 points which are very, very important: study of normal physiology and development, disease study and human models, biological products, for getting the biological products, vaccine safety and testing, and chemical and toxicity testing as well. Works? Cool? Chalo. Now, the last part of this chapter, that is ethical issues and biopiracy in biotechnology. So we studied about biotechnology in plants, we studied about biotechnology in animals, the uses of that, and now we are with ethical issues and biopiracy in biotechnology. So why there is a need for regulation of things? You know, if we are just there is no regulation for genetic modification and stuff like that, it can give rise to certain organisms which might not be really good. You know, coronavirus is said to be a product of genetic engineering itself. Okay, we never know that it might have been or it might not be true. There are so many things which are being produced which could be dangerous for us, which is why there is a need for regulation as well. So unregulated genetic modification can have unpredictable effects as well. Ethical concerns over the manipulation of living organisms, that is also there. Obviously, as me, as you, everyone thinks like we should not be harming monkeys, we should not be harming mice and things like that who have very much genetic resemblance to us. But so ethical concerns have also have to be taken up and to be thought about, which is why there is a need for regulation. Otherwise people will go around killing monkeys and doing this, so we do not want that to happen. Now regulatory body, who is the one who regulates all these things? So it is Genetic Engineering Approval Committee, GEAC. The abbreviated form will come, you might be asked to put down the whole sentence. So it is Genetic Engineering Approval Committee. So for genetic engineering, whatever approval is there is a committee being made. Okay, they only approve it. You remember like that. Genetic Engineering Approval Committee. It approves GM research and ensures what? Safety, my dear children. Now, patents and traditional knowledge. Company patent genetic materials and bioresources developed by indigenous people. you know what company actually there was a problem being done, that is in what do you call it, in a US company. Okay, so US company, what they did was they patented basmati rice, you know, basmati rice or basmati rice, whatever you say, it was patented. But it is grown from India, right? It is from our indigenous people who grow it, but they took the patent as in you do not have the right to then produce it and things like that. That is what you mean by patency. So can they actually do that? No, they cannot actually do that. We'll come on to that, but let me tell you the scenario. So companies patent genetic materials and bioresources developed by indigenous people, as in the people who are there from the, from own countries. Okay, now example, in 1997, a US company patented basmati rice derived from Indian varieties, my dear children. Other examples, patents on turmeric and neem for their medicinal properties. This was also been done, but can you actually do it? No, you cannot do it because there is some rules and regulations happening in our country and also in the worldwide which we have to follow, which is why the patent had been removed. Obviously, you cannot do that to anyone. Now, biopiracy is something that we should be knowing. What do you mean by that? Unauthorized commercial use of bioresources and traditional knowledge without compensation. So, this was being put on to them. Okay, biopiracy was being claimed for this particular act that they did. That is, they unauthorizedly used our resources. Okay, without even compensation, they put patent on that. Obviously, you should be giving compensation to the people who actually make it, that is the Indian farmers and things like that. You get it? So, biopiracy is very much important. Patency as in basically you have no right to discover it, produce it in any more level. There is only one company who can do this. That is what you mean by patency. If I have a patency for something, only I can do this. Okay, only I am allowed to do this, you are not allowed. That is what I mean. And what is biopiracy? Basically, this is tested. The unauthorized commercial use, if it is there, okay, unauthorized commercial use is there, I am basically utilizing, used to discover something and I am basically utilizing it for and telling that I did it. So without compensation, I am doing all these things. So this is what biopiracy is. So commercial use of bioresources and traditional knowledge without compensation is biopiracy. Now there are few terms given in your NCERT. It is developed countries. What do you mean by developed countries? Financially they are rich but poor in biodiversity. Developing country as in biodiversity rich but vulnerable to exploitation, my dear children. Okay, not means, it means that, okay, these are the things that is associated, not very much important, so you don't have to put too much into it. So preventing biopiracy, how we can do that? Some nations are creating laws to protect their bioresources. Okay, so Indian Patent Bill amendment includes provision for patent, emergency situation and R&D, research and development support. So Indian Patents Bill is there to protect us from all this patency that could be coming around. So obviously any nation which is there, any country which is there, it cannot put on patent for anything like that. There are certain laws which we have to abide. Okay, so Indian Patent Bill amendment is for us, my dear children. This is a bill that was made, and this is a very important thing. What was the amendment thing? So it was Indian Patent Bill amendment which was put down, and it is for the provisions for patents, emergency situations and R&D support. Just give me a second, there is a number also of the bill. Just give me a second, okay, I'll just check through and come. Yeah, so basically the second amendment of the Indian Patent Bill includes all this thing. Please remember that as well. So that question can be asked. What amendment? Such a second amendment that came in PU2, so anything can happen here in KCET as well. Okay, so this you can exclude. Rest of the things are really important. Please remember biopiracy and patent. Okay, works. And I think we are done with the whole chapter, starting from tissue culture. We went to somatic hybridization, then we came to are there any way for better crops, that is genetically modified crops, what are the uses of it, golden rice, vitamin A rich, and then BT crops, the cryogene and everything, RNA interference that is seen in tobacco plant infected by Melodigoin incognitia, then biotechnology in medicine where we spoke about insulin, my dear children. Okay, Eli Lilly was the company that was there. In gene therapy, it was adenosine deaminase deficiency, deaminase deficiency. Okay, so three things were there. Either you do bone marrow transplantation, or you do enzyme replacement therapy, or you do gene therapy, but only one cure is there, that is basically if you do changes in the embryonic cell, that is fine. Otherwise it is not. Then early disease diagnosis, you have PCR for amplification of even trace amounts of things, okay. Then enzyme and immunosorbent assay for basically and which works on the principle of antigen antibody binding. Then we basically spoke about transgenic animals, what is the use. We spoke about alpha-1 antitrypsin used for treatment of emphysema, and also one more thing, that is alpha-lactalbumin, that is a human milk protein being produced by the transgenic cow, that is ROC, okay. And then the ethical issue, GEAC stands for Genetic Engineering Approval Committee, my dear children. Patency is there, and then biopiracy is there, which basically says there is unauthorized, whatever unauthorized things are being said, okay, unauthorizedly using unauthorized commercial use of all the bioresources and everything without compensation is known as biopiracy. And to tackle that, in India, the second amendment was put down for the Indian Patent Bill, which provides provisions for all this, for dealing with all the issues that comes around it. Okay, so I hope this is clear, and with this we are completing biotechnology and its application chapter. I hope you understand each and every part of it, but still, if you have any questions, please feel really free to ask me. I'm not gonna do anything to you, my dear children. You can put out the questions as such, also I can give you the answers. If you're facing difficulty in that, first of all try and then tell, okay. And rest of the things, I think it's all fine, and do let me know if you're liking the videos, and like, share and subscribe. Wishing you all the best. Take care, not be tensed. Bye bye.